Good afternoon, everybody. My name is Mike Espy, and I'm a program director with the National Cancer Institute. And it's my pleasure to introduce Jodi Nunnari, today's WALS lecture speaker. Jodi grew up in Cleveland and studied pharmacology at Vanderbilt. She did her postdoctoral training at UCSF with Peter Walter, where she fell in love with the structure and function of organelles. Jodi has been a pioneer in the study of mitochondrial morphometry and continues to be a champion of cell biology. She is the chair of the Department of Molecular and Cellular Biology at the University of California in Davis. She's a member of the National Academy of Sciences. Congratulations, Jodi. And a past president of the American Society for Cell Biology. In this capacity is through ASCB that I first met Jodi, and we collaborated over the last 5 years in putting in the ASCB annual meeting and embedded symposia on emerging studies on mitochondria and cancer. And it's been a real hit with the community in merging the fields of cell biology and cancer cell biology. Fun facts about Jodi, she loves horses and she loves whiskey. So without further ado, we really look forward to Jodi's talk today on mitochondrial behavior.
Yes, he knows me well. I do love whiskey, and I have a slight horse addiction. We're currently at 3 horses in my family. They're a very expensive habit. So I'm really happy to be here today and share the work we do in my lab with you. We're really interested in mitochondria. When I was a postdoc in Peter Walter's lab, I worked on the endoplasmic reticulum and kind of hit a roadblock in my studies there on what was called the ribosome receptor then, and it turned out to be the translocon. And I just decided to switch directions, and I fell in love with this organelle, primarily because I became fascinated by the fact that it has its own chromosome and what were the rules there in terms of chromosomal transmission. And that led to just describing the compartment. And on this first slide here is a human cell in culture, one of the model systems we use. We're card-carrying cell biologists. I'm also, by the way, editor-in-chief of the Journal of Cell Biology. For anybody who wants to tell me about their latest and greatest story, I always have open and eager ears. And we use cells as a model system to kind of uncover fundamental principles. So I apologize in advance to those who are doing more relevant work in tissues. This just gives you an idea of probably what happens there. And this is the big picture question that has driven the work in my lab, which is how is mitochondrial behavior controlled inside cells? And what that boils down to is defining the machines on a molecular scale that function at this compartment, which you see here in blue, to give it this characteristic in this particular cell type. This is a U2OS cell in culture. This characteristic distribution and overall look. It's very tubular, very connected. And over the years in my lab, we have defined the machines that work on this organelle, such as dynamics, division and fusion, the proteins that do that job, active mechanisms to position the mitochondria in certain places where they're needed. We've recently begun to work on how they move around inside cells. And in hand, as we all know, structure-function relationships, we really are interested in how this compartment is put together in its very detail, and I'll talk about that towards the end. But really, this big picture question boils down to how are all these machines that we've identified and kind of delved into mechanistically, how do they function together to do the job that you see here? And the bias that we've had in my lab is that they do so with the goal, hopefully, of transmitting the chromosome in that compartment. And if you notice here in red, there's another star of the show. That's the endoplasmic reticulum and how it looks inside these cells. And one of the punchlines of my talk today is that these two fundamental eukaryotic defining compartments. So my worldview is that everything else can be made de novo except these two, and of course the plasma membrane. How do they collaborate together to govern the behavior of mitochondria? And again, here's my worldview. Right here, here's a very, very simple tree of life on this planet. And there's 3 kingdoms. Everybody knows this. And here we are, eukaryotes. We're pretty special. Our cells can become specialized. We can walk and talk and do all those great things. And that happened right here at this bifurcated point here in the tree of life. And this is right where we acquired mitochondria. So I like asking people, how many people believe in extraterrestrial life out there? Raise your hand. That's so hopeful. I love that in people, that hope. So Nick Lane, who thinks a lot more about the evolution of life than I do, and in particular the role of mitochondria, said if you were to meet a Martian, an extraterrestrial being, they would have mitochondria. So that's kind of how fundamental they are. And why is that? Because they're another compartment, and they do the work of a lot of metabolism, including energy production, and have allowed us to become way more efficient and specialized, hence eukaryotes. But a lot has happened since we acquired mitochondria, and I think people generally agree that mitochondria came from a bacterial ancestor that had probably about on the order of 4,000 genes. In human mitochondria, there's only 37 of those genes now. So a lot has happened since then. So a lot of these genes have been lost to the nucleus or lost forever, probably also driving evolution in that way. But the mitochondrial proteome, as we know, is much more complex than just 37 components. It's made up of around 1,100 proteins. And so a vast majority of the proteins that comprise mitochondria come from nuclear encoded genes. And so during this process of evolution, a lot has had to happen for these compartments to become what they are now, including the ability to import proteins, the nuclear encoded components that I just talked about, and also their lipid components. So mitochondria have 2 major biosynthetic pathways for phospholipids, for phosphatidylethanolamine, and the signature lipid in this compartment, again, very similar to their bacterial ancestors, cardiolipin. These are really 2 defining functional lipids for these compartments. They can't make them by themselves. They need the precursors for these lipids that come from the endoplasmic reticulum. And that's the first hint that these 2 compartments collaborate extensively. And all those machines that I talked about briefly on that first slide give rise to the behavior of this compartment are new inventions, by and large. And together, these kinds of machines allow the compartment to be built, of course not de novo, but to continue to be built, and also integrate with other pathways in the cell that are extramitochondrial. Signaling goes back and forth between the mitochondrial nucleus, for example. So the story I'm going to tell you today is how we've learned how these machines are connected, we think. It's a working model of how pathways that couple the various disparate machines are linked together to give a mechanism to transmit mitochondrial chromosome within the cell. And this is another model system we use. It's budding yeast, Saccharomyces cerevisiae. We like it because it's an in vivo system. It also has fabulous genetics, but mostly because it's in vivo, where we, by and large, ask similar questions. And so we tend to go back and forth in my lab. And while some of the components aren't necessarily stringently orthologs of one another, the strategies seem to be conserved with how cells get the job of mitochondrial chromosome transmission done. Using this simple system, my lab and a couple of other labs many years ago discovered the genes that encode the proteins that regulate directly mitochondrial division and fusion, and those are listed, the proteins are listed on either side of this equation. And so much like in the human cell that I showed you in yeast, mitochondria are also very connected into this tubular network in so-called wild-type cells. But you can disrupt this network by disrupting division and fusion selectively. So if you block fusion, division goes on and this connected network disconnects. And on the other side of the equation, if you block division, fusion goes on and the connections even get more connected into this net-like structure that you see here. And that wasn't surprising to us because when we describe this system, it turns out the rates of these events of division and fusion are roughly equivalent in this cell type. And so you can tip the morphology of the organelle, how it looks inside cells, by just altering the relative rates of division and fusion. And one of the major phenotypes that we noticed in these cells was that if you disrupt dynamics, the transmission of the chromosome, the fidelity of that process goes down. If you block fusion, you get complete loss. Division increases the rate of loss. And together, it described a picture of what the function of dynamics really is inside cells fundamentally. And it's not that fusion's important or division's important, because they are, but what really we learned is that what's important is they both exist together. So you have to have one and the other. And together, these processes maintain the function. You need mitochondrial DNA. They encode proteins involved in OXPHOS, oxidative phosphorylation, respiratory chain, and distribution. And that's most apparent in these cells here, where if, for example, you focused on this cell on the top, this is a full cell projection, it would be bare. There would be no mitochondria. And the way yeast work is they position their mitochondria at the cortex, because they don't really probably know where the bud's going to form, where they have to distribute. So you need dynamics to maintain function and distribution. And not surprisingly, if you disrupt these processes in human cells, it results in a lot of different types of human diseases. So it was kind of cool that when these genes were cloned that were required for division and fusion, the heart of the machines we discovered belong to a single protein family, which are called dynamin-related GTPases. And I had the opportunity to collaborate with Jenny Hinshaw here in the audience. And this is a micrograph that she took, I don't know how long ago, Jenny? A while ago, of the prototypic member of this family called dynamin-1, which functions during endocytosis and the scission of clathrin-coated pits from the plasma membrane. And these are large GTPases. They don't function like switches, but they're mechanochemical enzymes that self-assemble into structures like this that are associated with membranes. So they can do the work of membrane dynamics through assembly and assembly-stimulated conformational changes that are mediated by hydrolysis of GTP, so-called power stroke. And for mitochondrial division, there's a single one that lives in the cytoplasm that gets targeted to mitochondria. I'll talk about that in a second. And in fusion, the dynamins are distributed one on each membrane, and we were able to show by reconstituting fusion in vitro that they each control the fusion behavior of the membrane that they inhabit, and you can separate mitochondrial outer and inner membrane fusion, interestingly. So given that the division system was pretty simple, we did a lot of work over a number of years reconstituting the process of division and learning a lot about its mechanism. And this is just a summary of some of the work we did, again, a lot of it in collaboration with Jenny, where the two non-dynamin members that are required for mitochondrial division are responsible for targeting the division dynamin to the mitochondrial membrane. And then one of those players, this protein called MDV1, then functions as a positive effector of dynamin assembly. So it's like, okay, you're here, you're at the right spot, assemble into a helical structure that in a GTP-dependent hydrolysis manner mediates a power stroke for scission. And the then postdoc in my lab, Marin Ford, solved the crystal structure of dynamin, which turned out to be a really good model for how the mitochondrial division dynamin work, where you get G domains interacting across helical runs to produce the power stroke. I'm not going to go into the details. But we did a lot of work on this, and it was really cool. I enjoyed the mechanistic work, and it was great. But something always bothered us, and that was that all the machinery, whether you're in a yeast cell here, which I just told you about, or in a mammalian cell, which by and large doesn't have conserved receptors or effectors on the outer membrane, no matter what, they all are on the outer membrane. So we could never find anything inside the cell, inside mitochondria, that helped division. Drove us crazy. And so we often wondered then, what in the world is going on in terms of division site placement? So let's go back now in evolution to the bacterial progenitor of mitochondria and talk a little bit about how they divide. They use a GTPase also, but it's more related to tubulin called FTSZ. And they use a mechanism called the MIN system and a nucleoid occlusion mechanism system, two parts, that say the MIN system oscillates from pole to pole, and it says, don't assemble at the pole, this machinery, and only assemble in the middle. And the reason for that, of course, is that you're segregating the chromosomes here, and you want each of the daughters to get one. That's the whole point. You fast forward here, I told you already, there's nothing on the inside, but there are in eukaryotes, certain eukaryotes, a hybrid system. And if you look at this system that often retains the MIN oscillatory system and FTSZ, it's also acquired an outside dynamin. And so the initial work of constriction is done by this FTSZ mechanism, and then dynamin gets targeted to kind of finish the process off. And if you look at the morphology in these systems, these mitochondria are pretty discrete, bacterial-like, in terms of their morphology. And so a mechanism to place the division apparatus that relies on the ends kind of makes sense. But in the mitochondria I showed you in yeast and in human cells, the ends have lost their meaning, essentially. It's so heteromorphic, they're not a very good reference point. And so what then decides where division occurs? And the answer to this came in the form of a collaboration with Gia Volz and my then postdoc, Laura Lackner, who just got tenure at Northwestern. I'm so happy about that. Where here's a movie that Laura took, where now you're looking in green at the endoplasmic reticulum, and red are the mitochondria. And if you focus here on this little ER tubule that seems to be close by to mitochondria here, and you watch it for a while, that's where division's going to take place. This blew my mind. Here's a video from Gia Volz and her then graduate student Jonathan Friedman, who came and did a postdoc with me. He's now at UT Southwestern, so I'm really made out in this. And you can see the same is true here, where the ER and mitochondria come close together is where a future division site will happen. So it's not an inside mechanism. It appears to be an outside mechanism. And so, of course, being the mechanistic types, we wanted to know what the molecular basis was. And at the time, work from Benoit Kornman, who was in Peter Walter's lab, identified the first real prototypic tether between mitochondria and the ER. And it's a so-called aromatase complex, ER mitochondrial encounter structure. And what's important here on this slide is to know that there are integral ER membrane proteins as a constituent of this complex and integral outer membrane proteins. And intrinsic to this complex is the ability to transport lipids. And in fact, I think it's becoming clear that a more generalized function of contact sites between organelles is to be a hub for lipid transport. And Will Prince does a lot of that work. He's here. We've collaborated with him. He's awesome. Also, a component of this, and this is also might be a theme emerging, is this protein called MDM10, which also moonlights in the biogenesis of mitochondrial, protein biogenesis. And so here you have a cassette that does lipid and protein biogenesis. Quite interesting. And then an ancillary but really tightly associated component of this is the yeast ortholog of a protein called miro, which is really important for moving mitochondria around in mammalian cells. It hooks up to adapters that in turn hook up to the actin and microtubule cytoskeleton. But here in yeast, mitochondrial transport is all actin-based, and we really don't know what the hookup here is. But what's interesting is that this is an integral component of this contact site. And so I wouldn't have talked a lot about this complex if I couldn't tell you that when you look at where this complex resides, and it has a very characteristic focal appearance. So now we're looking at MDM34, I believe, as a component of the aromatase complex. They all look like this. This is where the ER and mitos come into contact with each other. So if I had another color to show you ER, this is published, which we did, it would be right here. And if you do a line scan across mitochondria, right here, you see this is a matrix marker. These mitochondria are already constricted at that point of contact. And this is prior, again, in other experiments, to the recruitment of the mitochondrial division dynamic. So you first get somehow this ER-linked mitochondrial constriction, which we still don't understand the mechanism of, maybe actin-related, based on others' work. You then get DRP targeting here once you get some constriction of the mitochondria. You get scission of the mitochondria, and then the tips move apart. And this is a very characteristic process. And it allowed my then student, Andy Murley, to order functionally the components I just told you about in this pathway now. So now we have a pathway that's centered around a contact site where aromatase mediates the contact and perhaps some of the constriction. We're testing that. You get targeting of the division dynamic by a mechanism I described. And then this motility step, the separation of the daughter tips from one another, is dependent on GEM1, which is the miro ortholog. So now we've coupled mitochondrial division to motility. And if you go back to the system again, the whole functional purpose of this is all great, in bacteria is to segregate the chromosome and to transmit it. And so we asked a similar question. And this is work we did in collaboration with Peter's lab, who developed this marker for nucleoids YME2. It's not important what it does. It just marks all the mitochondrial chromosomes in yeast, which we call nucleoids. And now the little dots that you're seeing are the chromosomes, not contact sites. And if you follow them, the nucleoids themselves, you see that division occurs in a manner that's spatially linked to where they are in about 80% of the cases. Not 100, but 80%. And so this has led in my lab to a very simple model where the contacts, the whole function of the contacts marking division is to link the distribution of mitochondria and mitochondrial DNA. And so we wanted to test whether this was true in human cells, whether this whole mode of distribution is conserved. And using, this is done by my postdoc Samantha Lewis, who's awesome. And the reason is not like a Me Too, and for the sake of academics. Rather, there are many components here that aren't conserved. Aromatase is not conserved. The mechanism of replication, mitochondrial DNA replication, is completely different. Nucleoid organization is different. Yeast are different than human cells. So we thought this was really important. And what I'm going to tell you is very robust in the sense that we have seen this process occur in a number of different cell lines in culture. And it was very challenging because mitochondria, unlike yeast, are more heteromorphic in human cells. You can't keep track of all of them at once, which drives me crazy. The ER is much more pervasive and articulated, and there are many, many, many more copies of mitochondrial DNA on the order of 2 orders of magnitude, sometimes more. So this was challenging, and so the first thing Sam did was just to describe the system, and we do this a lot. in a quantitative way. She looked at where the nucleoids are, here in green, marked by a protein called TFAM, which is basically the chromatin of mitochondrial DNA. And she looked at where those were relative to where regions of the endoplasmic reticulum come in close contact with mitochondria, or potentially. And this is just a line scan, and I'll just describe the quantitative analysis she did, which was that by cross-correlation coefficient, there was an extremely high probability that if you're a nucleoid, you're going to have some ER tubule very close to you. So maybe it wasn't that surprising then that when she looked at mitochondrial division relative to nucleoids and relative to the ER, she observed that very similar to the yeast system, in about 80% of the cases, mitochondrial division linked to where the ER contacts are is going to happen in 80% of the cases next to a nucleoid, or in close proximity to a nucleoid. And I'll just remind people that mitochondria have two membranes, an outer and an inner, and the nucleoid's in the very innermost compartment, the matrix, okay? So while you're thinking of this. And also very similar to yeast, although I didn't tell you this, is that sometimes you get nucleoids close to both ends from division, and sometimes only one end. And when you only have it linked to one end in segregation, that's the end that goes with the ER. So there's some real stable connection there that we think. The other thing she noticed, this was done independently. If you just look at nucleoids in human cells in culture over time, a lot of them have a lot of short-range motility, but there's a subset that move quite long distances, relatively speaking. And retrospectively, if Sam went back and took a look at what those were, they were products of mitochondrial division. And so now, this should feel kind of similar to what I've told you in yeast. Again, the strategy is being conserved, that the ER is serving to link the division and motility machines together, we think, probably by creating a microenvironment that clusters, for example, the motors, or allows the targeting of the division machinery right there. So one thing that kind of started driving us crazy, though, was, okay, that's great, but how many contacts, ER mitochondrial contacts, are there in a cell? So if these really mark division sites, are they all dividing? What's going on? And so Sam took a step back and took confocal data like this of the ER and mitochondria, thresholded it, and marked regions where they co-localized as potential regions of contact between the two organelles. And then used the great equalizer of time to determine which were stable. And we defined those as real contact sites. And I can tell you that a lot of these co-localizations weren't stable. And so for people who want to study membrane contact sites, it's not cool to use fixed data and co-localization to quantify regions of contact, because a lot of them aren't in contact. They're probably just close by. Okay, this is the problem with light microscopy. So there's a lot. There's over 100 per cell that are stable regions of stable co-localization. And if she looked at those regions, she really couldn't see anything happening to a majority of those. And by what I mean is events linked to division. So about 10% of them had, of these contacts had regions where the mitochondria were constricted linked to them. And then during the 5-minute period, about 1% of those converted into bona fide scission events, just by the process as I described to you before. And so what this told us is that, yes, ER mitochondria contacts mark future sites of division, but they're not rate limiting for division. And so I love kicking the can down the road. We do that a lot in this project. And so the question then becomes, what determines division site placement beyond the contacts? And so our logic was, well, the contacts, we think, are spatially linked to nucleoids. I told you that already. So is there something inside, really? And are all the nucleoids the same? Are they part of the process? And so we looked specifically at the fate of nucleoids that were undergoing replication, because I guess that's how you know you're alive, you replicate. So to do that, we had to develop a real accurate in-cell mark for replicating nucleoids for live cell imaging. And we turned to an accessory subunit of the mitochondrial DNA polymerase, a POLG2, based on the work from a colleague, Bill Copeland, who showed that this GFP fusion was functional on a biochemical level even. And here we are using the analog EDU to monitor incorporation of nucleotides into mitochondrial DNA, and I'll just blow this up for you and show you that nucleoids that are incorporating, that are replicating, are also marked with the accessory subunit of POLG2. So we thought we had a good marker. And in fact, if you do a comparison of EDU labeling of nucleoids versus POLG2, they're the same. And that's, of course, in contrast to the total population, which I told you already we could mark with TFAM, or there's a dye called picogreen that you can use. So at any snapshot in time, only a small fraction of the total mitochondrial DNA genomes in a cell are replicating, about 10%. And this makes it a really hard system, but fascinating to study, because it's not a very stringent transmission system like the nucleus, which, the whole cell cycle is devoted to. So about 10% at once. It's asynchronous with each other. It's asynchronous with the cell cycle. It is what it is. So we use this marker, and this was like this is one of my favorite outcomes of an experiment in my career. It turns out that replicating nucleoids are the best marker we have for future sites of division. Not the contact sites themselves, but the replicating nucleoids linked to the contact sites are. And the numbers are here. If we look at mitochondrial divisions stringently marked by the division dynamin DRP1, out of the total number, about 80% of these are linked to this POLG2. And if you recall, that 80% number is what I told you we got from looking at TFAM-labeled nucleoids, at the total population. So I guess, in other words, if you're going to divide at a nucleoid, you're dividing at a nucleoid that's undergoing replication. And that accounts for 80% of the division events in these cells in culture. So they're a real determinant of future division sites. And so this combined is a real pathway, we think, that's orchestrated by the membrane contact site, where the contact site is there. The nucleoid fires for replication inside the mitochondria. There's about an hour that happens here, an hour elapsed, and then you get these successive events of constriction, division, and motility. So it's this pathway, we think, for mitochondrial DNA transmission. And so what really blew us away was addressing the role of the ER in all of this, because the contacts are there. And so we did this by manipulating the steady-state structure of the endoplasmic reticulum, which is present in both membrane tubule-like structures and sheet-like structures. And these have separate proteomes. This is work by Junji Hu, who nicely showed this. They're structure function, man. They're functionally specialized regions of the ER. That's appreciated. And you can, much like in mitochondria tipping the balance of dynamics, you can tip the balance of the steady-state distribution of the ER membrane in sheets and tubules. So if you overexpress a protein called CLMP63, this is from work from Tom Rappaport's lab, you deplete the tubules and expand the sheets. And conversely, if you overexpress a reticulon, this is a tubulus shaping protein, RTN4A, you deplete the sheets and proliferate tubules. And if you co-overexpress them at roughly the same levels, you come up with a cell that looks kind of wild type in a nice distribution. And this serves as a nice control. So we did this and asked, okay, what's the status of mitochondrial DNA replication? Amazingly enough, if you deplete the ER tubules by overexpressing CLMP63, mitochondrial DNA replication plummets. And we think that it's more correlated with tubular ER for a couple of reasons. Double overexpression doesn't do that. So it's not CLMP63 overexpression per se. Rather, it more tightly correlates with loss of ER tubules here. And these are two measures of mitochondrial DNA replication. And the other cool thing about these experiments is they were mosaic on an intracellular basis, where when you overexpress CLIMP63, you could never quite deplete all the tubular ER. And in fact, the mitochondrial DNA genomes that were replicating that we could see in these cells were always associated with ER tubules and no place else. So again, this really tight correlation between that. So there's something about these contact sites that are really important for licensing the replication of mitochondrial DNA. So you need the ER to get replication, which I think is pretty profound. Has a lot of implications in tissues and things like that. Interesting things to test. So there's a lot of questions now that we're addressing. As I said, how are these contact sites seem to integrate machines into functional pathways? How does that happen? And how does this tubular ER license mitochondrial DNA replication? And we think that this is intimately related to the question of how the cells know and count the number of mitochondrial DNA genomes they have. It's a relaxed system. Replication is, woo. How does the cell keep track of it? Because they're not wildly varying in terms of copy number. So how do the cells count? So I'm going to tell you a little bit of our unpublished data on this, where we've been asking to address this question about the crosstalk between the ER and the known components involved in mitochondrial DNA replication. So the mitochondrial DNA replisome, beautiful work from Maria Falkenberg, has been reconstituted in vitro, at least the minimal system. And it's comprised of a polymerase, which I've already told you a little bit about, that has a trimer of 2 accessory subunits and the polymerase, single-stranded binding protein, and this helicase called Twinkle that's derived from phage. So evolution is wild. We're asking about these components specifically and their relationship to ER mito contact sites and trying to now order their assembly. And I can tell you in our hands, Twinkle, and this is work also done by Anu Somalainen in mice, Twinkle seems to be the rate-limiting component for mitochondrial DNA replication. It's the only component that we can overexpress that increases the number of mitochondrial genomes undergoing replication. So just have that in your head. So we look then at Twinkle, and lo and behold, Twinkle marks replicating nucleoids. That's really great. But we noticed that a majority of the Twinkle foci don't localize with nucleoids that are replicating as marked by EDU and Pol G2. So there's a fraction of more. But if we look at all the Twinkle foci, we notice that like the nucleoids, because they're localized to nucleoids, they're at mitochondrial ER contacts also. And so we kind of surmise, and now we're doing live cell experiments, that Twinkle probably loads on to the nucleoids before the polymerase does, which would make sense if it's rate limiting. But what we really noticed that kind of puzzled us at first was that there was a significant number of Twinkle foci that didn't co-localize with mitochondrial DNA. It was still at ER mitochondrial contacts, but it didn't co-localize with mitochondrial DNA. So they were just kind of out there, these Twinkle foci, these structures. And this is just with anti-DNA antibody, and this is endogenous Twinkle. So we go back and forth with slight overexpression that doesn't alter copy number and looking at native proteins too. And so that prompted us to ask the question, well, what is the role then, in terms of the replisome assembly, of mitochondrial DNA at all? Is there a role? And so to do this, we created cells that lack mitochondrial DNA and co-cultured them with cells that contain mitochondrial DNA and looked at these components again. And maybe not surprisingly, TFAM, Pol G2, mitochondrial SSB lost that focal appearance that's characteristic of the nucleoid because that's where the DNA is packaged. So they can no longer bind to the DNA because the DNA is not there. Now they're diffuse in the matrix. Fine, that makes sense. But look here at what Twinkle does. Twinkle remains punctate, and this is just a blow-up within the mitochondrion, and that's just the quantification of that. And in fact, endogenous behaves the same way, and the number of Twinkle foci don't vary too much in the presence or absence of mitochondrial DNA. So the numbers are roughly equivalent. So what are these structures? Well, I can tell you that they're also at mitochondrial ER contact sites. They're all enriched there. And if we do the same type of manipulation of the ER, they really require the tubular ER for their stable formation. So they're at mitochondrial contacts, and they require these contacts to form. And that's just reminiscent of the behavior of replicating nucleoids, so a nucleoid that has mitochondrial DNA and is replicating. So there's very tight correlation here. And this just struck us because going back to the yeast system, as I told you, they replicate differently. Probably more recombination-driven replication. Human mitochondrial DNA really don't recombine. There's a component called MGM101 that's a Rad52 ortholog that in row 0 cells is also focal, maintains its focal distribution. And look at how nicely aligned it is with the aramase complex. Again, MGM1 assemblies are on the very inside, aramase is on the very outside of mitochondria, but these are clearly spatially linked with one another. And we think, our thinking is shaped a lot on what we think contact sites do. They transport lipids, as I said, and we know, based on lots of published work, that they're domains, though they have their own proteome kind of themselves, okay? And so based on this, we think that they are microenvironments to mediate the assembly of things like of Twinkle, for example, inside the matrix somehow, because of their specialized composition of lipids and proteins. And so based on that, we began to ask whether any of the replosome components cared about lipids at all. And this is just our preliminary work showing that Twinkle, and this is in collaboration with Maria Falkenberg, who is the expert in making these proteins, does have an affinity for acidic lipids like cardiolipin and PA. And not surprisingly, all these lipids are the precursors that come from the ER, and PA gets funneled into the signature cardiolipin lipid here. And so we asked whether in the context of a membrane, a liposome that we made in vitro, whether this is also true. And Twinkle, yes, does interact selectively with liposomes that contain about 2 times the amount of cardiolipin that is normally in the inner membrane, consistent with perhaps a concentration in a microdomain. And Twinkle itself is a very interesting helicase. At steady state, it's already assembled into the characteristic donut shape that these helicases have, but it has a primase domain similar to the T7 phage that it's derived from, but it doesn't function in doing primase work. Rather, it's a domain that kind of gets assembled on the top of it via this linker region. And so we've done a lot of structure-function analysis to determine what are the important determinants in Twinkle that allow it to interact with the membrane, and all our work is consistent with this region, this linker region here, that somehow helps that primase domain load on and get it going. And when we look at this linker region, these are mutations that abrogate the binding that are in this region. Other mutations in the helicase domain don't, for example. If we look at this linker region, there's a pretty short but very classic amphipathic alpha helix here. And these are known for interacting with membranes and even in some cases shaping them. And then consistently, this little piece of Twinkle is both necessary, obviously, and sufficient to allow it to interact with membranes in vitro. And I implied this, but this is just showing you data that amongst the known replosome components, only Twinkle has the ability to bind lipids. So we don't see this for SSB, for TFAM, for POLG. And now we've done it in all different combinations, and Twinkle's still the only one that binds to these lipids. And so we're left now with a simple model. It's of copy number control, where again the contact sites are microdomains. This is a model that allow the assembly of this Twinkle-like replication platform that's rate limiting for the replication of mitochondrial DNA, and it's present in a finite amount. So the copy number then is not magically controlled. It's controlled by the number of ER mitochondrial contact sites, these domains, and the amount of Twinkle that's expressed inside mitochondria and the assembly of the structure, the dependence of that on the assembly of the structure. So this is a model that we're testing, and overarchingly, it really tells you that the ER in this context is a system regulator that allows all these machines to coordinate, including replication of mitochondrial DNA and its transmission. So it's seen as a system regulator. And so I want to switch gears a little bit. I hope I'm not going to overstay my welcome and tell you a very short story, because I think it's profound in regards to this. So I told you that we're really interested in mitochondrial ultrastructure. So you're trying to imagine the matrix while I'm talking to you, but this is what it really looks like. This is a tomogram that Terry Fry took and reconstructed, and you see that this is the inner membrane, and it's very articulated. Everybody knows that they're called cristae, right? And they penetrate into the matrix. So that's what is going on here. And there's domains within the inner membrane. So you have an inner membrane that's continuous. It's one of the most protein-dense membranes in the cell, and that has to laterally differentiate, much like the ER structure of tubes and sheets. This also is laterally differentiated into what we call a boundary region, which is the membrane close to the outer membrane, and these cristae structures. And what this has started us thinking about, because these are the metabolic hubs, is now back to the tree of life, how metabolism itself drives the organization, the self-organization of this membrane. And we've worked on how this membrane gets put together. We know that phospholipids are really important, in particular the signature ones I told you about, to differentiate this membrane. The respiratory complexes themselves play a role, like ATP synthase dimerases, and is enriched on the curved edges of these cristae. There's a complex that we and others identified that's called micose that we think determines the copy number of cristae and their distribution. So we know something about it, but we really still don't know how proteins sort into these domains. And so to approach this, we simply inventoried the domain localization of a bunch of intermembrane proteins, and we turned back to our yeast system to do this. So there's a problem. Everybody has hit it at one point who does microscopy. This is a known boundary protein, TIM50, and a known cristae protein. And if you now look at their localization within this mitochondrial tubule at a line scan, oops, that's a little weird, but you can see that they're not resolved at all, right? But we know that they are by EM. We know this. So you can do super resolution immuno-EM. It's pretty low throughput. So we did a riff on expansion microscopy where you can take this aromatase complex and create a degron allele of the cytoplasmic component, MDM12, and the morphological change that happens as a result of loss of this contact is that the tubular ER collapses into a pretty large sphere. And if you take a look at that sphere now at the same markers, just focus here, a boundary and a cristae marker, now we can resolve those because it's just making mitochondria big. So the mitochondrial proteome, this is beautiful work from Klaus Fahner's lab, in yeast has been nicely annotated and characterized based on mass spec. A majority of proteins are in the inner membrane. It's very dense. Out of those proteins, we could inventory about 300 of them. They're functional GFP fusions that are nicely expressed. And this is the distribution we get. A majority are in the cristae. There are some in the boundary. This other category is the one, of course, that my lab became interested in because they're kind of perverse like that. They represent proteins that are targeted to multiple organelles. This is interesting. Some that are not enriched in either compartment. It's really emphasizing there must be active mechanisms for this. And then the other category is proteins that formed their own domains, not boundary, not cristae, but something else. They included the mucose proteins, which we knew because we could visualize those in wild-type mitochondria, but also a cluster of proteins that are responsible for the biosynthesis of this really ancient lipid CoQ. And for this, we teamed up with experts in this field, Dave Pagliarini and Josh Kuhn. So what did we see? Here are the cluster of CoQ proteins we saw. So again, not boundary, not cristae, but rather they form these discrete domains that are found within these spheres. We know they're intermembrane associated. And coenzyme Q is an extremely greasy lipid. I mean, look at the mammalian version. It's huge tails that get placed on heads that do the redox chemistry that's necessary to move electrons within the electron transport chain and pump protons. It's essential for respiration. So how does it get made? It's a fascinating pathway. It uses isoprene units that are derived from the mevalonate pathway, which is also responsible for synthesizing sterols. And these isoprene units, along with the precursor head group tyrosine or 4-HB, get transported into mitochondria. Still mysterious how that happens. And the first thing that happens is these tails get polymerized by CoQ1 that can somehow count them, do 6 in yeast and 10 in mammalian cells. And then the head gets attached to the tail. And then the rest of this pathway, all these other things, successively modify the head to do the redox biochemistry. So imagine this. A lipid gets made, and it's in the membrane, and this machinery has to, somehow find that lipid and modify the head group of it. And some of these proteins are enzymatic and have enzymatic growth. Others do not. For example, this CoQ8 is thought to be a lipid chaperone that kind of pulls on the head, say, to allow accessibility. Anyway, so we asked many questions. One of which is, what do these proteins look like in normal-looking mitochondria? And we saw that neither the tail polymerase nor the head-to-tail attachment had any special domain localization. And these are all functional. But strikingly, what we saw was all the head modifiers localized to very discrete regions that we could identify. I hope you can see these here. Even within a normal tubular mitochondria, we had the resolution to do that. The non-enzymatic, some of them did, some of them didn't. I think it's particularly interesting that this lipid chaperone didn't. It's probably surveying the entire membrane for head to tail, for CoQ intermediates. So when they form these domains, they're all together in these domains. So the domains, as far as our analysis goes, all have the same composition of these head-modifying enzymes. And we can determine the copy number relative to one another, for example. We're determining relative stoichiometry using a standard. This is a kinetochore cluster protein of known numbers. And these are quite big. So we estimate there's like 180 copies of CoQ9 in these clusters. These are big. And of course, the machinery, because it's an ancient lipid, is strictly conserved between yeast and humans. And so we looked in human cells, and by and large, this is a validative antibody against native CoQ9, which is a head modifier. You can see that, as in yeast, they form these domains. And so what's required for domain assembly? Even though CoQ1 and CoQ2 are not themselves components of these domains, they're required. And in fact, any essential component in this pathway to make this lipid is required. It's an all or none thing. Doesn't matter where you are, what step you mediate in the pathway. If you're essential and you delete that, the gene for that enzyme or that protein, you don't get domains. And that's just shown here. The non-essential ones, you still get domains. So what we know is that these CoQ, we call them domains, is tightly correlated with a functional pathway. And it's all or none, extremely highly cooperative. You either get them or you don't. And that, again, correlates with production of product. And that really suggests this role for the lipid itself, the intermediate lipid. Again, this idea that the chemicals and their fluxes and their interactions through the metabolic pathways is driving self-organization. And this kind of goes with data from Kathy Clark's lab, who's also a pioneer in this field, where it doesn't, again, matter where you delete. Even if you delete a component, like, for example, 5, which is downstream, you only accumulate this very first product of head-to-tail attachment, all or none, highly cooperative. So to ask whether the substrate itself, the CoQ intermediate lipid, is required, we just did a little dead body experiment where this allele of CoQ6 is inactive, and you can bypass its function by adding this compound called vanillic acid. So you skip over the enzymatic function of 6, and you go right into 3. The body is required, though, to get this to work. And so here are yeast cells that harbor this allele of CoQ6. In the absence of vanillic acid, there are no domains. If you add vanillic acid, voila, the domains form. We've also done the reverse, which is to shut off flux through this pathway, and the domains disappear. So it's very clear that domain formation and production obviously are tightly correlated. And we believe that the CoQ substrate itself is required to form these regions of concentrated head-modifying enzymes. And we don't think it's de novo synthesis or anything fancy like that, because if you look at total cell fluorescence in the absence and presence of vanillic acid or by Western, the protein levels don't change of these components. Rather, they take what's there and they get reorganized into these assemblies, which is pretty cool. So this is pretty interesting. We think that domain formation and the number are really sensitive biomarkers for flux through this pathway, which could be useful for other people's work. They're not dependent on mitochondrial DNA. We're looking genome-wide at what they are dependent on beyond what I've told you. So they're standalone structures, very similar to the Twinkle that I told you about previously for mitochondrial DNA replication, which is why when we looked at where these domains were positioned, I guess we were not that surprised that they're positioned by ER mitochondrial contact sites. So we have in yeast a very good marker for all the ER mitochondrial contacts. It's this protein called LTC1 that transports sterols at these sites. It's not important what it does really for the purpose of this. And you can see that where the contacts are, we get very good co-localization of the CoQ domains inside. Again, this contact being outside, the domains being inside, it's another way to it's another organizational center. In this case, it's a metabolic pathway. And if we disrupt the two types of contacts present in yeast, this aromatase complex I told you about, or this LTC1, the domains are incredibly disrupted. We no longer, they decrease in their numbers, and in fact, they tend to coalesce, so their distribution is completely screwed up. So the domains are, seem to rely on contact sites also for their formation. So the purpose of creating such a thing, we think, is to maintain the substrate accessibility. So again, going back to the fact that the lipid is such a grease ball, if you let go of the head, how are you ever going to find it again? So you have to create a really processive system, and that's, I think, what these domains are for. You wouldn't want any old CoQ intermediate hanging around. And this has a name. Other people call them metabolons. And here's another example of the de novo purine nucleotide biosynthetic pathway that's been described in the literature, where the enzymes form these clusters of proteins, sometimes in proximity to mitochondria because they share substrates through this pathway. So these metabolons are really important. And they're not just for CoQ biosynthesis. We've also found them for isoleucine and valine biosynthesis, for the TCA cycle, for pyruvate dehydrogenase, and we're now putting them together in this framework of how they all relate to one another. So I guess that reiterates the point that mitochondria contacts are really system regulators for assembly and for distribution and to create pathways inside cells. So I'll end. The work on the nucleoid I told you about in human cells is done by Samantha Lewis. The work on CoQ is done by Kelly. And Sam was helped out by a postdoc, Marina, and a former undergrad, Lauren, on the work in collaboration with Maria Falkenberg and Dave Pagliarini and Josh Kuhn. So I'm happy to take questions. Thank you for your attention.
The FAS is going to have a reception in the library after this, but we have time for a few questions.
Everybody's
It's a little bit over.
Very, very fascinating talk.
Thank
you.
Thank you.
It's very close to what I'm doing as far as biology of mitochondria in relation to immunobiology of cancer. My question is, or maybe it's not fair to ask, but to propose. Do you, for example, if there is an activated cell which has a lot of mitochondria like macrophages, do you see some differences when you activate or in a resting status of the immune cells like macrophage M1, M2, whether there is the connection between ER and mitochondria would change, and or whether the chronic stimulation would disturb even the replication of mitochondria?
That's a great question. We've only worked under these homeostatic conditions that I've described. I'll just make one short comment on that, which is that this system might be tweaked from cell type to cell type. If different things become rate limiting, that's definitely a possibility, but certainly any perturbation of the ER mitocontacts I would predict would have pretty dramatic effects on mitochondrial DNA replication. So,
very, very good.
Thanks.
Good subject.
Thanks.
So yeah, that was beautiful. You showed that eliminating ER tubules with a CLMP63 knockout overexpression caused what did it cause? A decrease in mitochondrial DNA replication.
Yes.
So is it unidirectional, like from the ER in? Like what happens to division or contact sites in row 0 cells? Or in Twinkle knockout cells?
Yeah. So don't know about Twinkle knockouts yet. We're doing those experiments now.
That might be the key, right?
Yes. So we're doing those experiments now. I can tell you our preliminary data suggests that the contacts are still there in the Twinkle cells.
Oh. Okay,
so I think it's kicking the can a little bit, yeah. But there's something that are at the very top of the chain.
And the last quick question, because it's changed over the years. Currently, what's the idea on the number of DNA copy numbers per nucleoid?
So I think we did some STED microscopy ourselves, work from at least 2 other labs, David Clayton and Nils Larson. Consistent with that, in these cells in culture, they're solitary.
Okay.
Yeah.
Thanks.
They're single copied.
Great work. I was wondering if we could get your thoughts on the potential contribution of mechanical strain or tension at the mitochondrial membrane that might be driving fission, and this would be consistent with the ER model and the actin model, and potential the density of nucleoids also changing the strain at the membrane that might drive that. And also, is there any evidence that a similar type of model might be contributing to the protein domain formation on membranes, where there's potential membrane sensing capabilities of proteins that might drive domain formation?
Wow.
Sorry. So many questions.
I think that's I think it's the most recent work doing that beautiful high-resolution actin assemblies. It's like, what are they, purse strings, right? That must be involved in that initial constriction step. We haven't done any work ourselves related to that, but I do like the idea. I like simple models based on reality. I tell my students in postdoc, I just went to the Biophysical Society meeting. I'm a big ASCB person, right? And I said, oh my God, I came back from this meeting. So you go to ASCB and you learn how things might work, and you go to the Biophysical Society meetings and you learn how they really work. And that's because of all the physical parameters. So, you know, we're not doing any of that, but I think it must play a role. Like, for example, why does motility happen after division? It must be clusters of motors that are needed to generate the force, things like that. So I can't really...
Yeah, no, it's great, thank you.
We're getting the heave-ho, so I think...
Oh, we are? We're getting heave-ho.
So I'm afraid we're going to have to close the session and take up the questions one-on-one in the library.
Who's heave-hoing us?
Thank you.
Thank you.